A mark, release, recapture experiment was organised in March 2018 in a 20-hectare area located in Carnaíba do Sertão, Brazil. During a period of 2 weeks, 215,800 marked sterile male Aedes aegypti mosquitoes were released and monitored for the next 4 weeks. This required the production of more than 700,000 adults in the mass rearing facility of Mosquitoes in Juazeiro, located a 20-minute drive from the release area. Sex separation was achieved daily at the pupal stage using mechanical Hock sorters, thanks to the size difference between sexes, with females being bigger than males. Sterile males were marked with different colours of day-glow fluorescent dust within a cold room at 5 degrees. The next morning at 6 a.m, sterile males were compacted into canisters that were placed in the cold box at 10 degrees for transport to the release site. The drone took off from a football court located in the middle of the village. Releases were conducted from the centre of the football court for point releases, or following pre-programmed release lines for path release. Between each flight, a cold canister containing up to 50,000 sterile male mosquitoes was loaded onto the drone. Canisters were maintained within an icebox at 10 degrees prior to loading. In the case of point releases, the drone undertook a stationary flight at an altitude of 50 or 100 meters. Point releases were conducted with different colors and compared to a ground release. Mosquitoes are released by a rotating cylinder, where they are transferred from the cold canister, maintained at 8 to 10 degrees, and subsequently fall onto a chute, where they are dispersed by the airflow. Following point releases, recapture rates of mosquitoes decreased with altitude, contrary to their average dispersal, which increased with release altitude. The survival of the 3 groups was similar. For path releases, the drone followed the release lines spaced 80 meters apart, with mosquitoes released over the entire treatment area at a density of 2,500 to 5,000 per hectare. Post-release monitoring was split into 4 sectors, where traps were collected daily by different teams simultaneously. Adult mosquitoes were collected using BG Sentinel traps, baited with BG lure, and thereafter brought to the lab for identification. Following path releases, marked mosquitoes were recaptured in 24 out of 35 active monitoring traps, which indicated a uniform release pattern over the whole study area. Their densities were strongly correlated with those of wild males, allowing a homogeneous ratio. After these releases, the sterile to wild ratio peaked at 0.8, and the rate of non-viable eggs upon 0.5, showing that the irradiated sterile males mated with wild females and were sexually competitive against wild males.